Who will present something on interpretable deep learning.
Hello everyone, my name is István. I'm a machine learning scientist at Computomics. And we'd like to start by, does this work? I would like to start by introducing you to the people involved in this study from the company down below here, and we are also part of the ARC Center of Excellence for Plant Success, which gives me the opportunity and the privilege to work with Mark Cooper and Owen Powell. We have seen a lot of evidence to the fact that machine learning is an indispensable tool for predictive breeding, because even general-purpose machine learning regression models are really good at capturing complex gene-by-gene and gene-by-environment interaction. But one can go beyond that, go beyond off-the-shelf machine learning models. And this is what we do at Computomics by designing and deploying machine learning models that were built for the purpose of predictive breeding. And today I'm going to show you an example of that through a series of experiments that follow this working hypothesis here. That by incorporating information about the underlying biological gene-to-phenotype network into the architectural design of these machine learning models, we can expect to gain in key areas. We expect to have better predictability, better performance stability under data drift that is caused by the breeding program, by the selection, and very importantly, have better interpretability. So we are going to use artificial data because here we have the liberty to freely tune some key properties of the data and also have a very robust, well-known ground truth, so we have a fair performance evaluation of the model. This is the backbone of the data set, this model biological network for shoot branching, where there are 40 QTLs related to the endpoint trait, time to bud outgrowth, that is under direct selection. And there is a metabolite network in between. 3 hormones and sucrose are related to groups of 10 QTLs in an additive way, and then they interact. So for example, auxin promotes rigolactone and inhibits cytokine. And we use this network, this model, to generate data. And for that, we use Owen Powell's own breeding program, which is an in silico breeding program simulation, where environments are fixed, so no G by E for now. And he also simulated 400 SNPs. Some of them are markers of these QTLs. So we start with 1,000 F2 genotypes. We do an intensive selection of top 10%. So we select the top 100 performers, do random crosses to get 1,000 new genotypes in each selection cycle. And we do this for 10 cycles in 5 replications, which are different from each other in terms of random effects. And we select for fast bud outgrowth. We have 2 series of experiments with high and low heritability of 0.8 and 0.5 in the endpoint trait, with a corresponding signal-to-noise ratio of 20 and 15 for the intermediate traits. We develop our predictive machine learning model on replication 1 on the early cycles, using either SNPs or QTLs as genetic input features, and using the true values of the 4 intermediate traits and the final trait as well. And at prediction, the model only takes as at input either SNPs or QTLs and then predicts all of these traits. So I'm showing you the data that we are working with. For cycle 1, the distribution of the intermediate and endpoint traits in solid lines, and already the next cycle with the dashed line. And here below, I'm showing the input genetics, 400 SNPs that were projected down into 2 dimensions using the UMAP algorithm. And I'm going to show you the data drift across these selection cycles. So as we are progressing and aggregating newer and newer data from these cycles, we can see that the newer cycles always drift away. And this is very strong, particularly for cytokinin, but also for strigolactone and the endpoint trait. Here in the genetics, you can also see that drift clearly. Okay? Now, by the time we reach cycle 5, we have already aggregated our development set that we will use to build a machine learning model. And then we are going to deploy that to predict on these consecutive test cycles. These are shown in red, the test data. And you can see that I'm also showing these gray curves, what would happen if we aggregated all the in-between cycles into our data set. We would still be only chasing the runaway distribution of the test cycle. So there's a huge data drift because of selection, and the machine learning model will basically have to extrapolate. Here in the genetics, you can also see that by the time we reach cycle 10, our target set is completely detached from the original locus of the development data set. This is a very challenging problem, and actually we are not going to aggregate these cycles. We are going to stick with the first 5 cycles just to make it more interesting and more challenging for the model. So we're going to develop a neural network that is based on these interactions. We start with, as an input, either the QTLs or the SNPs, and then we have 5 branches for these 5 metabolites, where we have feature selectors that will learn which of these features are important in an additive way for these metabolites. And then these dot products model the additive genetic effects. Then we have some interaction branches where we explicitly tell the model what interacts with what. So for example, with strigolactone, we tell that it interacts with auxin and so forth. And we model these interactions, the result of the interactions as additive corrections on top of the additive genetic values. And here the model is going to output for each sample 4 values, the 4 total genetic values that we can compare to the real values, forming 4 losses there. And then on the top of that, there is an interaction branch which describes the dependency of the output of the endpoint trait on these metabolites, forming the 5th loss. So we have 5 losses in total there. We implement this with TensorFlow, and for the baseline, we use an XGBoost regressor model. That's a very versatile general-purpose regression model that takes in only the genetic features, QTLs or SNPs, to directly predict the time to bud outgrowth. We fit the model by minimizing the RMSE loss and tune the hyperparameters to optimize R-squared for the validation set using Bayesian optimization algorithm. So I'm showing you the results which are Pearson correlation on the endpoint trait as a function of the test cycle. So predicting in this many years into the future for all 5 replications. Remember, we trained the model on replication 1, and we are predicting for all the other applications with that. So we can see a very consistent performance gain by our metabolite INET neural network with respect to XGBoost, and we have very good performance stability over these prediction cycles. We can also use the model to infer feature importances for QTLs and for SNPs that I'm going to show you right now. So here there are the QTLs, and these bars show feature importances extracted from the XGBoost model for the high heritability experiment. And these dots show the true values, so our ground truth that we compare against, true effect sizes of these QTLs. You can see that XGBoost does not get the complete picture here. There's an exponential decay of these. However, if we take the 4 metabolite branches and investigate what are the values of these feature selectors, then we get this. So this is the bars are learned feature importances for the QTLs, and the dots show the real values, the real effect sizes. You can see that these correlate very well, And also, the metabolite INET learned to separate the additive QTLs from the indirect non-additive QTLs per metabolite. How about SNPs? This is a Manhattan plot. The red bars show learned feature importances for the SNPs by our baseline model, the XGBoost. And these green bars show the ground truth locations of the QTLs. You can see that the model identified plenty of the QTLs, but it's a fuzzy picture. There's a lot of noise, and most of the QTLs are missed. What happens if we use a neural network? The neural network, we are going to look at the QTLs by metabolites. So the green bars show only those QTLs that are additively related to auxin. The others are shown in gray. And you can see that the learned SNP feature importances are aligned in an excellent way with these QTLs. This is true for all the other traits as well. So we effectively identified all the relevant genes, almost all or relevant QTLs per metabolite using this neural network. We can also investigate the model to get further insight about these feature interactions between the metabolites. Once we have trained the network, we can isolate a subnetwork here and give it to a SHAP analysis. So here we are going to use this and these layers as input, and we can perform a SHAP analysis to get mean feature importances. So here, mean absolute SHAP values are shown. And this way, we can not only rank the importance of the features, but we can directly compare the importance of auxin with respect to the additive QTLs of cytokinin. We can see that the importance of auxin is comparable to the combined effect of all additive QTLs of cytokinin. We can also plot SHAP values for each sample as a function of the value of the intermediate trait. And we can see clearly an anti-correlation with auxin and the correlation with sucrose. And from this, the network effectively discovered that auxin inhibits cytokinin and sucrose promotes cytokinin. We can do the same thing with the final prediction branch, do a SHAP analysis on this subnetwork, get feature importances of the total genetic values for the endpoint trait, and plot these interaction plots from which we can infer that strigolactone increases and sucrose and cytokinin decrease the time to bud outgrowth. Finally, what would happen if we didn't know how this network really looked like? So I told you that we explicitly tell the model where these interactions are. So for example, we told the model that sucrose does not influence strigolactone levels. But what if we didn't know that? What if we assume this red connection here? So what we can do there is we can do this. We can train a model with this falsely assumed interaction there. And then, like before, isolate from the trained model this interaction submodel here, and use these layers as input, and do a SHAP analysis. So what we would get is that sucrose has a negligible importance here. So we can get feedback from the SHAP analysis and correct our model, do an ablation of this branch, and retrain the model with the correct architecture. The interaction of auxin and sucrose from this wrong connection here can be seen here. And what you can see is that sucrose basically mimics the dependency of strigolactone and auxin, but on a much, much slower scale. So what happens here effectively is that this dependency leaks through this wrong connection to the extent by which it can be fitted to the noise pattern in the data. This also indicates that this is a wrong connection, so you can get rid of it. And what I don't have time for now is that one can actually map out the complete architecture of this metabolite network doing this analysis iteratively from bottom to top, do SHAP analysis, so assume some connections, do a SHAP analysis, get feedback, ablate the unnecessary connections, and basically almost from scratch you can figure out the shape of this network, provided that we know that these 4 metabolites are involved in this process, and they are fully describing the endpoint trait, but not necessarily knowing how. So we have seen that our working hypothesis was correct, and we have gained a lot of things by incorporating biological knowledge, and this shows the potential of doing this for real-world applications. So we have achieved higher predictive performance, a robust model against data drift, with the ability to extrapolate even to unknown data domain. A model could separate additive and non-additive genetic effects. We could compute feature importances, understand the metabolite interactions, accurately identify groups of genes per metabolite, and even there's the possibility of mapping this biological network only based on data and a deep learning model. Thank you very much.
Given the time, I think we might have one very quick question.
Fred.
Oh. Felix.
Yeah, I was just wondering, this network, what is the time resolution at which these interactions occur? Because I mean, I had the impression that everything is static. I would say you're at the hormone level. So say these hormones react to each other, well, I don't know, on a second scale, minute scale, or not? Yeah.
No, this is a static model. Yeah, so we assume that so these are balanced, so these evolve through time and reach a stationary...
this is a stationary situation over a growing season.
Exactly.
Okay, no, because my worry was that actually you would need to model the gene expression in relation to, well, environmental stimuli. But I mean, this is on average how it would look like over the growing season.
Yeah.
Okay.
Yeah, this is just a showcase study that fits 15 minutes. But yeah, there's an unlimited amount of additional complexity.
It's realistic to think of this as a static average expression over the growing season. But can discuss that.
So let's keep it for the discussion.
A bit more crystals we'll talk about.